you had to do one activity for today that was writing down the names of the chromosomal abnormality. Was it your homework to write down? Ahora que lo he dicho, no estoy tan segura que fuese eso. Did I ask you to write down the names of... Ay, no te hemos oído. Otra vez.
Teacher.
Tell me.
¿Había que mandártela? Digo, yo tengo esta,
no.
pero no te la...
No había que mandármela. No, ni hablar.
Ah, vale.
Ya me lo escribiréis todo, me lo daréis todo cuando me tengáis que dar el cuaderno, pero así poquito a poco no quiero yo que me deis nada. ¿Pero qué era? Writing down the names, was it your homework? Or did I ask you to do the following activities? Aquí abajo.
Era el 9, el 10 y el 11.
Esto es, ahora lo veo aquí, ahora que lo veo ya sí lo veo. Eso es, sí. So, activities 9, 10, and 11, that was your homework. Because the chromosomal abnormalities, we wrote down the names as we saw the PowerPoint presentation. I launched the PowerPoint presentation yesterday evening, so you're supposed to be able to open it right now. In case you don't have the names, write them down here with the information in the PowerPoint presentation, but it was one by one in the same order, so that's not difficult at all to find those names. Activity number 8 was referring to the prokaryotic cells chromosome, and that was one activity that we had to do for Christmas, the description of the prokaryotic cells chromosome, explaining the difference between the chromosomes in prokaryotic cells and eukaryotic cells. So that was not your homework for today, but it was something that I asked you to do before Christmas. So I guess I've got one volunteer somewhere to answer this question. Dime, ya la acabo de hacer, la acabo de ver y ya está aquí con nosotros, ¿verdad que sí, Sara?
No veo la pantalla.
Ay, gracias por avisarme, que siempre se me olvida hacer esto y luego os tengo aquí, ¿ves? Ah, sí, ahora mejor, ¿verdad? Bueno. Alguien que me ha hablado por aquí es Ana, ¿verdad? Ana, describe the prokaryotic cells chromosome and explain differences between the prokaryotic and the eukaryotic chromosome. Do you have anything written down in this question number 8 as an answer?
Yes.
Yes, you do. What do you have? Vamos a ver si podemos hacer esto. What do you have?
In prokaryotic organisms, all the genetic material is packed into a single DNA molecule.
Hold on. One important thing is that prokaryotic organisms only have one single molecule of DNA. That's very important. Okay, go on.
The DNA is in the form of a double helix, which forms a closed ring with no free ends.
Okay, so another important thing about this prokaryotic chromosome is that everything is in the form of a closed ring with no ends. Okay, in the case of eukaryotic cells, we're going to have longer threads, but they are not closed as a ring. They're going to form threads of chromatin that when they condense, they have this shape of an X, like the chromosome that we've seen, the metaphase chromosome that we've seen. So important differences. One of them is only one molecule of DNA. Second difference, that is a closed ring. Any other thing? That is... Ana, ibas a decir otra cosa más, seguro.
No, solo tengo eso.
But you can also say that it's permanently attached to the plasma membrane, which is what we've got in the theory. So it's very important that you mention that is a single DNA molecule and that is a close ring with no free ends. Okay, that's the most important difference. While, because you've got to explain eukaryotic, while you can say that in eukaryotic cells we've got more than one molecule of DNA. In fact, Ana, queda bien delante del público. In fact, in humans, how many molecules of DNA do we have in each nuclei?
I don't know.
Yes, you do. How many molecules? How many chromosomes?
46.
46. The number of chromosomes is the same as the number of molecules of DNA in the nucleus, okay? So in bacteria, it doesn't matter the species of bacteria, there's only one molecule of DNA in forming the genetic material. But in the case of the rest of the living things, algae, fungus, protozoans, animals, plants, whatever, we've got more than one molecule. We've got more than one chromosome in its cell. How many? It depends on the species. We said that it's very important that you don't consider that the higher the number, the more evolved that you are. Sometimes it could seem so because we've got 46 and we've got here, for example, that mice only have 40, that this famous fruit fly, I'm sure that you've seen fruit flies at home. Have you ever seen this fruit fly? At home, this tiny fly with these red eyes. A ver si tengo una en... Ay, que me salen todas haciendo guarrerías. En real size. A ver. For you to see the size. Well, I'm sure that you've seen these flies. Sometimes these flies are very famous in genetics because they only have 8 chromosomes. ¿Dónde estábamos? Aquí, ¿verdad? They only have 8 chromosomes, so it's quite easy for scientists, or it's easier, at least it's easier for scientists to analyze their genome. And they've been used a lot to study how the characters are inherited. So this animal, Drosophila melanogaster, is very famous in genetic. We're going to talk about it a lot. So it seems like those animals that are not very evolved, they have less chromosomes, but it's not true. We've got a yeast, a type of yeast, the one that we use to produce beer, that contains 32. And we mentioned the dog as an animal containing 78 chromosomes. So the number is not closely related on evolution. It's just a number of molecules in the nucleus. We've got 46, and we were talking about the previous day that these 46 chromosomes could be classified as autosomes. We've got 22 pairs of autosomes because they don't determine your sex, and sex chromosomes that in the case of humans, in the case of mammals, is X and Y chromosomes. If we have all of them homologous, all of them 2 by 2, then you are a girl. And if one pair is not exactly the same size, one is bigger than the other, the sex chromosomes, one of them is bigger than the other, then that means that you are male. I said that this happens in mammals because, for example, birds, they work in the opposite way. In the case of birds, if the two sex chromosomes are identical, we've got a male bird. But if one of them is bigger and the other one is smaller, then that is a female bird. Okay, so that's a different way to determine sex. In the case of mammals, them two being identical, double X means female. In the case of birds, because they don't work in the same way, instead of X and Y, we call them Z and W. And double W means, Z and W means that is girl, female bird. Okay, so that would be the answer for this one, number of and type. Question number 9 was your homework. We've mentioned loads of times the difference between chromatin and chromosome. Sarah, are you there? Can you tell me what's the difference between chromatin and chromosome?
I don't have it.
You don't have it, but you have some ideas. And if you don't, maybe having a look at a picture that represents chromatin and the same picture representing a chromosome, that could give you an idea. Any idea on the difference between chromatin and chromosome?
The chromatin is made up of all the chromosomes.
They are made up of the same material. In both cases is DNA with proteins. H means a type of protein. So in both cases is DNA and protein that forms them. But when the cell is working, this DNA with proteins will be in the form of chromatin. When the cell is going to divide, chromosomes will be visible. So what's the difference between this and that? Just the shape, the organization. Chromatin is DNA and proteins are not condensed when the cell is working. When the cell is going to divide, then all the DNA is going to be condensed in the form of chromosomes. Okay, so chromosomes form when chromatin condenses. Then the other difference was between chromatin and a chromatid, two terms that are very similar, but you should not be confused with them. Chromatin again is the DNA and proteins that are not condensed when the cell is working, and you could add that a chromatid is one part of the one of these two identical structures that form the chromosome. One of the two identical structures that form chromosomes, and they are joined by the centromere. So chromatid is one of the two identical structures that form chromosomes. Very important that they are joined by the centromere. So we've got another term here, centromere, that is important that you differentiate from other terms that are centrosome and centrios. Podríamos añadir another activity here that would be the difference between centrosome, centromere, centriole. Okay, it's important that you differentiate these three terms that are so similar. Centrosome and centriole, organelles in the cell. Centromere is part of this chromosome. Centrosome is formed by two centrioles. Two centrioles, and the aster fibers form the centrosome. Those aster fibers form the, does anyone know what's the name of the structure that the centrosome is going to form, and it's very important for the division? ¿Cómo se llamaba eso? That is so important for the division. ¿Quién me ayuda? Tengo un micrófono abierto de alguien cobarde que no me quiere decir nada, y luego tengo cerrados los de los valientes que me teníais que decir cosas. This structure, what's the name of this structure?
The aster fibers.
Is the aster fibers, they form this structure, the mitotic spindle. Well, if you pay attention to this structure, the mitotic spindle, the fibers that go out from the centrioles, these fibers will join the chromosome exactly in which region? In the centromere. In the centromere, there's one group of chromosomes in what we call the kinetochore, pero esa última kinetochore, you don't need to know it. In the centromere, these aster fibers will be attached. So when the cell is going to divide, these two centrioles separate from each other. The aster fibers will pull, and then they will separate the two chromatids in the chromosome. So as you can see, these terms are very similar, but in order to make it possible for you to understand it, you've got to be able to differentiate them. So very important that you've got this activity and that you understand the answer of this activity. Centrosome, made of two centrioles and the aster fibers. Centromere, one region in the chromosome that joins the two chromatids. Vale. And the last one is centromere and telomere. And we've got someone here in this class. A ver, ¿qué? ¿Hay alguien que ha debido decir algo? No. Álvaro, que te veo por aquí. Álvaro, what's the difference between centromere and telomere, or what do they mean? Alguno de esos dos. Centromere. You don't have this answer, but do you have this picture in your photocopy? This one, or maybe this picture. You probably do. Okay, we've got this picture. I'm going to a little bit bigger for you to be able to see it. We've got a chromosome here, and there are different structures that we must know from this chromosome. The first of them is the one that we were mentioning right now. That is the centromere. Centromere is the region of the chromosome that keeps these two chromatids together. One chromatid is joined to the other chromatid by the centromere. We call this centromere as well primary constriction primaria, which is just one structure that is joining these two chromatids together. Centromere is the region in which the two chromatids join together. This is important because depending on where this centromere is located, we had different names for these chromosomes. Remember, in the photocopy you've got metacentric. No sé si esto lo llegáis a ver. Igual un poquito más grande no estaría mal, ¿verdad? Ah, qué bien. Así. Depending on where the centromere is located, we've got metacentric if both arms are the same size. Submetacentric, this one is smaller than that one. Acrocentric and telocentric, 4 names that you've got to know. Depending on where the centromere is located. Is it in the center? Is it a little bit higher? Is it on the last part? 4 different positions. 2 parts that we must know. But the question was asking you about this telomere. So what's the telomere? It's just the last region, the external region, the extreme, you can also say, the last part of the chromosome. Okay, you've got here that the telomere are the region of the DNA at the end of this eukaryotic chromosome. And they are very important to make the replication of the chromosome stable. They're going to protect the rest of the chromosome. And we said that they get shorter and shorter as we get older and older. That's the telomere. And I ask you to remember this part of the word telo because it means end. And we are going to mention that as a phase in mitosis. We are going to see mitosis today. Apart from this one that you had in the activity, so centromere is the region in which the 2 chromatids join. Telomere is the external region of DNA, the region at the end of the chromosome. You've got many different definitions. That would be this last part of question number 9. And in question number 10, dice Blanca que no puede meterse. In question number 10, no puede meterse, pero no puede ni siquiera esperarme en la sala de espera. Bueno, pues no pasa nada, luego que vea el vídeo y ya está, yo la perdono. In question number 10, you've got make a picture of a chromosome and label the parts. What do you have to do? Just this. Okay? In question number 10, just make a picture of a chromosome similar to that, and remember to include the chromatids, this long part, the telomere, the end of the chromosome, the centromere, the central part, and we've got these 2 terms as well, the P arm, that is the short one, and the Q arm, that is the long one. Remember that when we draw chromosomes, we always put the short arm in the upper part. But when you see them in the microscope, you're going to see something like this. So if I give you this chromosome, how do you know which one is the P, which one is the Q arm? Always the shorter arm is the P arm. This one is the Q. Every metacentric chromosome, every chromosome like the one that you've got here has 2 P arms, 2 Q arms, okay, forming these 2 chromatids. Well, those are important parts that you must know. Apart from this theory, a ver, si pudiera yo ya quitar división. Vaya, se ha ido lo que no es. 200. Apart from this part of the theory, what the bacterial chromosome is, we've already seen it. How are the chromosomes in eukaryotic cell? We've seen it as well. What are the structure of these chromosomes? Chromatids, the centromere, that is the primary constriction, but centromere is the important term. The arms, P arm and Q arm, remember that the structure with proteins is kinetochore, but it's not so important this year. Telomeres is an important term as well. Types of chromosomes, depending on the shape, they are also important. We mentioned the day before what diploid means. What are the homologous pairs of chromosomes? That's another very, very important term. Homologous chromosomes are chromosomes that one of them comes from your dad, the other one comes from your mom, and they code exactly for the same character, even though they may have different information. So if this one is coding the color of your eyes, this one coming from your mom is also coding the color of your eyes. One of them may say brown and the other one says blue, but they both refer to the same character, that is homologous. They have the same function, they code for the same information, and they are going to have the same size, the same pattern in colors. Okay, that's what we were doing the day before, putting them 2 by 2 in homologous pairs. Only diploid organisms have homologous pairs, because we've got in those pairs chromosomes coming from our dad and our mom. Okay, and we represent the number of chromosomes in diploid organisms with this N. N is the number of pairs that we've got. So in our case, in humans, our N is... ¿Quién es un valiente que me escribe aquí rápidamente? What's the number that our N stands for? How many pairs of chromosomes do we have? Un escrito rápido o un micrófono abierto. Venga. That's very quick. 23. Okay, 23, Aitana, not 22. 23, because we've got 23 pairs. 22 of them are autosomes, but we consider that we've got 23 pairs, so our letter N is 23. In the case of mice, their N is 20 because they have 40 chromosomes. And in the case of the fruit fly, their N is 4. From those 23 pairs, 22 are autosomes, somatic pairs, and only one of them is the pair of sex chromosomes. In the case of female, one pair. In the case of males, one pair. From this pair, one of them is X, the other one is Y. Then, who is going to determine the sex in humans? Is it the dad or the mom that is going to determine the sex? When we pay attention to this information, vamos a poner uno nuevo. Your dad in the sperm cell, your dad in the sperm cell had how many chromosomes? 23. Your mom in the nucleus of the egg had exactly 23. Okay, so how did your mom do this ovule? Well, in the ovary, your mom has cells with, in the ovary, cells with 46 chromosomes. In your mom, in her ovary, cells with 46. But these cells in the ovary are going to form ovules that have 23 chromosomes, only 23. The process by which your mom was forming these cells is known as meiosis. And we'll see it this week. This process is a division that happens in the ovary in order to form these ovules with 23. The same thing happens in the testicle of the male organism. Is going to form from cells that have 46 chromosomes, is going to form cells with only 23 chromosomes. That is going to happen there with another process of meiosis. This division that is meiosis. So your mom had all the chromosomes in homologous pairs. Every cell in the ovary is double X. Then in this division, what is the information that you are going to have? Is going to be an X, no doubt. This egg here is going to have an X as the sex chromosome. What about your dad? Your dad in these 46 chromosomes is going to have an X and a Y. So your dad is going to form some sperm cells like this one, that are going to contain in the nucleus an X, but will also form other chromosomes that are going to, other sperm cells, sorry, that are going to have this Y. All the ovules that your mom is producing, all of them have this X in the genetic information. But your dad is producing 50% of the sperm cells containing an X, 50% of the sperms containing a Y. So if this sperm cell, one of these millions of sperm cells that are containing an X, fertilize the egg, then it will form a girl. But if the sperm cell that fertilizes the egg is one of these type containing a Y, then the zygote will form a male organism, a boy. So who is really determining the sex of the individual? Is it the mom or is it the dad that determines the sex? Who determines the sex in mammals?
The father?
Is the father. Always the father, the dad, that determines the sex. Because depending on the type of sperm cells that fertilize the egg, then I'm going to either have a girl or a boy. Okay, that's going to be an important thing, this unit. Then it's easy once I do this karyogram, that is this figure, this picture. Once I have it, it's easy to know whether I have a boy or I have a girl. But this karyogram was also important to know whether there is any chromosomal abnormality in the baby that has been formed. And we mentioned, and you've got in the PowerPoint presentation, we've mentioned different chromosomal abnormalities like Turner, Pateau, Edwards, triple X, superwoman or XYY, different chromosomal abnormalities. And remember that it's important that you determine whether it's a boy or a girl, and the most important characteristics, but especially just being able to identify it. What are we going to see today? The last parts, how we transmit the genetic material from the parent cell to daughter cells. A ver, tengo aquí algo. Ah, no, eso no que me ha llegado esto. How we transmit it in your virtual class. A ver si soy capaz de llegar ahora, que esta no. In your virtual class, you've got here. Dime.
¿Cómo has dicho que se llamaba el proceso cuando los 46 se pasan a ser 23?
Este, dices?
Sí.
Meiosis. And that's what I was going to show you. We've got here the last part of the unit after chromosomes, the structure, the number, and the abnormalities. The last part of the unit is cell division, mitosis, and meiosis. And that's what we're going to see this week. We're going to see the process of mitosis and the process of meiosis, the difference between them and all the stages that are going to happen. But in order to be able to understand these parts, you must know all the names of the parts of the chromosome, and the centrosomes are their mission. Vale? That's very important that you understand this, centrosomes, the mission of the division, and all the parts of the chromosomes. And now we are going to see these two processes, mitosis and meiosis. What do we use them for, and what's the difference between them? Well, what do we use mitosis for? We've mentioned the mitotic spindle before. ¿Os acordáis que lo hemos dicho, lo acabamos de decir hace nada? Mitotic spindle. Mitotic spindle was this structure that was formed thanks to the centrosome and these aster fibers. When was this mitotic spindle formed? Well, if I see chromosomes there, it's because the cell is dividing. I only see chromosomes when the cell is dividing. So what is the mission of this mitotic spindle? Help in the division of the cell. What is the most common reason for the division of the cell? If you go back to this first presentation, why do cells divide? The most common reason for the division of my cells is increasing my size. You that are growing are making a lot of cell division, or replace cells that have got old. So you've got to replace them. Or if you make a wound, you've got to replace that. In order for you to do these two things, your cells must divide. This process of cellular division is known as mitosis, and it happens in different stages. But it's going to work, this division is going to work different when the cells are prokaryotic. Sara, me parece que te has dejado el micrófono. Eso es. This division is going to be different in prokaryotic cells. Because in prokaryotic cells, we are not going to see the chromosome with this shape. We are not going to see the nucleus disappearing because they never had a nucleus. They do it, this division, in a different way. Do they do it in order to grow or to repair structures? No, they don't, because we saw that unicellular organism, they only do it to perform reproduction. When their cells divide, is because the organism is reproducing. And every prokaryotic organism is unicellular. Acordaos que esto funcionaba en un sentido sí, pero en el otro no. Every prokaryotic is unicellular. Not every unicellular is prokaryotic, but every prokaryotic is unicellular. So when prokaryotic organisms multiply a cell, is because they are reproducing. They do it thanks to a process called binary fission. And what they do is the chromosome that we said it was attached to the cell membrane is going to start replicating in both directions. So at the end, I have something like 2 chromosomes that will just separate. The cell will elongate, will get longer and longer until it breaks. It's a very simple process that takes very little. In 20 seconds, you've got 2 new bacteria. Bueno, as an average, because it depends on the species. But you've got this process, binary fission for prokaryotic cells. When the cells have nucleus in eukaryotic cells, the division of the cell receives this name, mitosis. Lo digo porque one question in the exam could be a true or false question: bacteria reproduce using mitosis? And the answer is no. Prokaryotic cells do not use mitosis. Only eukaryotic cells reproduce using mitosis. So if the question is, protozoan reproduce using mitosis? Yes, protozoan organisms, they are unicellular. So if they do division, they reproduce, and they are eukaryotic, so they do the division with mitosis. Then that is true. Protozoan organisms reproduce using mitosis. But in the case of bacteria, when the cell divides, they reproduce. That's true. But they don't use mitosis because they don't have nucleus. They reproduce using binary fission. What we'll see the following day is different animations in which you will understand the whole life cycle of a cell. What they do when they are working, how they start the division, and how they finish with mitosis. But we are not going to see it today because it's 5 to 11 already. So I don't want you to do more than to see further things. I didn't ask you to do any activity, and we've got some things about theory por aquí. So questions number 11, 12, and 13 could be your homework for today, okay?